Multiple global crises are competing to destroy our way of life. The solutions, new antibiotics and vaccines, carbon removal, and alternative fuels are all produced using bacteria, but we don't fully understand our tools. I'm a genome engineer. I take small pieces of DNA out of cells to determine what they do. More specifically, I'm interested in minimal genomes. These are cells containing only the DNA content that's essential for the cellular survival. The reason we do this is we want to understand what's fundamental for life, but we also want to work out what pieces of DNA make cells faster and better able at producing the products we need to solve these crises. Genome engineers typically use both computers and lab work. They do this because computational work is faster and cheaper, and it means we can test our designs before we implement them in the lab. To do this, you need to have a model of your cell, an algorithm, which is basically just a recipe for solving a problem, and tools such that you can implement your work from the computer in the lab. I work with Mycoplasma genitalium. It's a parasite of your genitals. We use that for two reasons. One, it's the smallest self-replicating natural organism that we know of, which means it's already close to a minimal cell. And two, it has a whole cell model. That's a model that accounts for all of the DNA content of the cell, and that's super important for minimal genome research. So, we have a small bacteria, we have a model, and we have tools, but we don't have an algorithm. And that's where I come in. I created an algorithm that is very similar to Minesweeper, the game. Basically what we do is we take out small pieces of DNA from the cell, similar to pressing a tile, and see if that kills the cell, finding a mine. The first steps are easy. You break up all of the DNA pieces and assign it to a tile, and then you press each tile one by one. If that kills the cell and you found a mine, you lock that tile and prevent it from being pressed again. The next steps are harder. You found all the obvious mines, but some mines can be primed or deactivated by prior tile presses. These conditional mines can only be found by basically testing every other tile but them. So, the way we do this is we start pressing big groups of tiles all at once, and then combining those big groups that don't set off a mine. You keep doing this process until you can't really do any large groups anymore, and you lock those in place. You then take the remaining tiles and break them into small groups, and start matching them in patterns as well. You keep doing this process until you can't move any more tiles without setting off a mine. My algorithm managed to remove 35% of the DNA content of our computational cell. If this works in the lab, it will be a record breaker. It will be the smallest self-replicating organism we've ever produced. But more importantly, it might be able to be applicable to new bacteria and help us save the world. Thank you.